Jump to content. Main menu. Main menu. Move to sidebar hide. Navigation. Main page. Contents. Current events. Random article. About Wikipedia. Contact us. Contribute. Help. Learn to edit. Community portal. Recent changes. Upload file. Search. Search. Appearance. Donate. Create account. Login. Personal tools. Donate. Create account. Login. Pages for logged out editors. Learn more. Contributions. Talk. Contents. Move to sidebar hide. Top. 1. History. 2. Methods. Toggle methods subsection. 2.1 Animal studies. 2.2 Human studies. 2.2.1 Twin and family studies. 2.2.2 Measured genetic variants. 2.3 Quasi-experimental designs. 3. General findings. Toggle general findings subsection. 3.1 Genetic influences on behavior are pervasive. 3.2 Nature of environmental influence. 3.3 Nature of genetic influence. 3.4 Additional general findings. 4. Criticisms and controversies. 5. See also. 6. References. 7. Further reading. 8. External links. Toggle the table of contents. Behavioral genetics. 26 languages. Eisenbachenkar. Català. Deutsch. Eesti. Espanol. Esperanto. Francis. Bahasa Indonesia. Netherlands. Uzbeka / Polski. Portuguese. Suomi. Svenska. Turks. Edit links. Article. Talk. English. Read. Edit. View history. Tools. Tools. Move to sidebar hide. Actions. Read. Edit. View history. General. What links here? Related changes. Upload file. Special pages. Permanent link. Page information. Cite this page. Get shortened URL. Download QR code. Print / export. Download as PDF. Printable version. In other projects. Wikimedia Commons. Wikidata item. Appearance. Move to sidebar hide. From Wikipedia, the free encyclopedia. Study of genetic environment interactions influencing behavior. Genetic psychology redirects here. For Franz Brentano's concept of genetic psychology, see Genetic Psychology, Brentano. For the journal, see Behavior Genetics, Journal. Part of a series on psychology. Outline. History. Subfields. Basic psychology. Abnormal. Affective neuroscience. Affective science. Behavioral genetics. Behavioral neuroscience. Behaviorism. Cognitive / cognitivism. Cognitive neuroscience. Social. Comparative. Cross-cultural. Cultural. Developmental. Differential. Ecological. Evolutionary. Experimental. Gestalt. Intelligence. Mathematical. Moral. Neuropsychology. perception, personality, psycholinguistics, psychophysiology, quantitative, social, theoretical, applied psychology, anomalistic, applied behavior analysis, assessment, clinical, coaching, community, consumer, counseling, critical, educational, ergonomics, feminist, forensic, health, humanistic, industrial and organizational, legal, media, medical, military, music, occupational health, pastoral, political, positive, psychometrics, psychotherapy, religion, school, sport and exercise, suicidology, systems, traffic, concepts, behavior, behavioral engineering, behavioral genetics, behavioral neuroscience, cognition, competence, consciousness, consumer behavior, emotions, feelings, human factors and ergonomics, intelligence, mind, psychology of religion, psychometrics, terror management theory, lists, counseling topics, disciplines, organizations, outline, psychologists, psychotherapies, research methods, schools of thought, timeline, topics, psychology portal, V.T. E. Behavioral genetics, also referred to as behavior genetics, is a field of scientific research that uses genetic methods to investigate the nature and origins of individual differences in behavior. While the name behavioral genetics connotes a focus on genetic influences, the field broadly investigates the extent to which genetic and environmental factors influence individual differences, and the development of research designs that can remove the confounding of genes and environment. Behavioral genetics was founded as a scientific discipline by Francis Galton in the late 19th century, only to be discredited through association with eugenics movements before and during World War II. In the latter half of the 20th century, the field saw renewed prominence with research on inheritance of behavior and mental illness in humans, typically using twin and family studies, as well as research on genetically informative model organisms through selective breeding and crosses. In the late 20th and early 21st centuries, technological advances in molecular genetics made it possible to measure and modify the genome directly. This led to major advances in model organism research, for example, knockout mice, and in human studies, for example, genome-wide association studies, leading to new scientific discoveries. Findings from behavioral genetic research have broadly impacted modern understanding of the role of genetic and environmental influences on behavior. These include evidence that nearly all researched behaviors are under a significant degree of genetic influence, and that influence tends to increase as individuals develop into adulthood. Further, most researched human behaviors are influenced by a very large number of genes, and the individual effects of these genes are very small. Environmental influences also play a strong role, but they tend to make family members more different from one another, not more similar. History. Edit. Farmers with wheat and cattle. Ancient Egyptian art 1422 BCE displaying domesticated animals. Selective breeding and the domestication of animals is perhaps the earliest evidence that humans considered the idea that individual differences in behavior could be due to natural causes. 1. Plato and Aristotle each speculated on the basis and mechanisms of inheritance of behavioral characteristics. 2. Plato, for example, argued in the Republic that selective breeding among the citizenry to encourage the development of some traits and discourage others, what today might be called eugenics, was to be encouraged in the pursuit of an ideal society. 2. 3. Behavioral genetic concepts also existed during the English Renaissance, where William Shakespeare perhaps first coined the phrase nature versus nurture in The Tempest, where he wrote in Act 4, Scene I, that Caliban was a devil, a born devil, on whose nature nurture can never stick. 3. 4. Modern-day behavioral genetics began with Sir Francis Galton, a 19th-century intellectual and cousin of Charles Darwin. 3. Galton was a polymath who studied many subjects, including the heritability of human abilities and mental characteristics. One of Galton's investigations involved a large pedigree study of social and intellectual achievement in the English upper class. In 1869, 10 years after Darwin's On the Origin of Species, Galton published his results in Hereditary Genius. 5. In this work, Galton found that the rate of eminence was highest among close relatives of eminent individuals, and decreased as the degree of relationship to eminent individuals decreased. While Galton could not rule out the role of environmental influences on eminence, a fact which he acknowledged, the study served to initiate an important debate about the relative roles of genes and environment on behavioral characteristics. Through his work, Galton also introduced multivariate analysis and paved the way towards modern Bayesian statistics that are used throughout the sciences, launching what has been dubbed the statistical enlightenment. 6. Galton in his later years. The field of behavioral genetics, as founded by Galton, was ultimately undermined by another of Galton's intellectual contributions, the founding of the eugenics movement in 20th century society. 3. The primary idea behind eugenics was to use selective breeding combined with knowledge about the inheritance of behavior to improve the human species. 3. The eugenics movement was subsequently discredited by scientific corruption and genocidal actions in Nazi Germany. Behavioral genetics was thereby discredited through its association to eugenics. 3. The field once again gained status as a distinct scientific discipline through the publication of early texts on behavioral genetics, such as Calvin S. Hall's 1951 book chapter on behavioral genetics, in which he introduced the term psychogenetics. 7. Which enjoyed some limited popularity in the 1960s and 1970s. 8. 9. However, it eventually disappeared from usage in favor of behavior genetics. The start of behavior genetics as a well-identified field was marked by the publication in 1960 of the book Behavior Genetics by John L. Fuller and William Robert Bob Thompson. 1. 10. It is widely accepted now that many if not most behaviors in animals and humans are under significant genetic influence, although the extent of genetic influence for any particular trait can differ widely. 11. 12. A decade later, in February 1970, the first issue of the journal Behavior Genetics was published, and in 1972 the Behavior Genetics Association was formed with Theodosius Dobzhansky elected as the association's first president. The field has since grown and diversified, touching many scientific disciplines. 3. 13. Methods. Edit. The primary goal of behavioral genetics is to investigate the nature and origins of individual differences in behavior. 3. A wide variety of different methodological approaches are used in behavioral genetic research, 14, only a few of which are outlined below. Animal studies. Edit. This section needs expansion. You can help by adding to it. February 2020. Investigators in animal behavior genetics can carefully control for environmental factors and can experimentally manipulate genetic variants, allowing for a degree of causal inference that is not available in studies on human behavioral genetics. 15. In animal research, selection experiments have often been employed. For example, laboratory house mice have been bred for open field behavior. 16. Thermoregulatory nesting. 17. And voluntary wheel running behavior. 18. A range of methods in these designs are covered on those pages. Behavioral geneticists using model organisms employ a range of molecular techniques to alter, insert, or delete genes. These techniques include knockouts, floxing, gene knockdown, or genome editing using methods like CRISPR-Cas9. 19. These techniques allow behavioral geneticists different levels of control in the model organism's genome, to evaluate the molecular, physiological, or behavioral outcome of genetic changes. 20. Animals commonly used as model organisms in behavioral genetics include mice. 21. Zebrafish. 22. Drosophila. 23. And the nematode species C. elegans. 24. Machine learning and AI developments are allowing researchers to design experiments that are able to manage the complexity and large data sets generated, allowing for increasingly complex behavioral experiments. 25. Human studies. Edit. Main article, Human Behavior Genetics. See also, Heritability and Quantitative Genetics Pedigree Analysis. Some research designs used in behavioral genetic research are variations on family designs, also known as pedigree designs, including twin studies and adoption studies. 14. Quantitative genetic modeling of individuals with known genetic relationships, for example, parent-child, sibling, dizygotic and monozygotic twins, allows one to estimate to what extent genes and environment contribute to phenotypic differences among individuals. 26. Twin and family studies. Edit. Main article, Twin studies. Pedigree charts showing an inheritance pattern consistent with autosomal dominant transmission. Behavioral geneticists have used pedigree studies to investigate the genetic and environmental basis of behavior. The basic intuition of the twin study is that monozygotic twins share 100% of their genome and dizygotic twins share, on average, 50% of their segregating genome. Thus, differences between the two members of a monozygotic twin pair can only be due to differences in their environment, whereas dizygotic twins will differ from one another due to genes in addition to the environment. Under this simplistic model, if dizygotic twins differ more than monozygotic twins, it can only be attributable to genetic influences. An important assumption of the twin model is the equal environment assumption. 27. That monozygotic twins have the same shared environmental experiences as dizygotic twins. If, for example, monozygotic twins tend to have more similar experiences than dizygotic twins, and these experiences themselves are not genetically mediated through gene-environment correlation mechanisms, then monozygotic twins will tend to be more similar to one another than dizygotic twins for reasons that have nothing to do with genes. 28. While this assumption should be kept in mind when interpreting the results of twin studies, research tends to support the equal environment assumption. 29. Twin studies of monozygotic and dizygotic twins use a biometrical formulation to describe the influences on twin similarity and to infer heritability. 26. 30. The formulation rests on the basic observation that the variance in a phenotype is due to two sources, genes and environment. More formally, VAR, P, equals G plus backslash display style var p equals g plus g backslash times backslash epsilon plus backslash epsilon, where p backslash display style p is the phenotype, g backslash display style g is the effect of genes, backslash display style backslash epsilon is the effect of the environment, and g backslash display style g backslash times backslash epsilon is a gene by environment interaction. The g backslash display style g term can be expanded to include additive, a2 backslash display style a caret 2 dominance, d2 backslash display style d caret 2 and epistatic, i2 backslash display style I caret 2 genetic effects. Similarly, the environmental term backslash display style backslash epsilon can be expanded to include shared environment, c2 backslash display style c caret 2 and non-shared environment, e2 backslash display style e caret 2, which includes any measurement error. Dropping the gene by environment interaction for simplicity, typical in twin studies, and fully decomposing the g backslash display style g and backslash display style backslash epsilon terms, we now have var p equals a2 plus d2 plus i2 plus c2 plus e2 backslash display style var p sad smiley a caret 2 plus d caret 2 plus I caret 2 plus c caret 2 plus e caret 2. Twin research then models the similarity in monozygotic twins and dizygotic twins using simplified forms of this decomposition, shown in the table. 26. Decomposing the genetic and environmental contributions to twin similarity. 26. Type of relationship. Full decomposition. Falconer's decomposition. Perfect similarity between siblings. 1.0 equals a2 plus d2 plus i2 plus c2 plus e2, backslash display style 1.0 equals a caret 2 plus d caret 2 plus I caret 2 plus c caret 2 plus e caret 2. 1.0 equals a2 plus c2 plus e2, backslash display style 1.0 equals a caret 2 plus c caret 2 plus e caret 2. Monozygotic twin correlation, RMZ, backslash display style r underscore mz. RMZ equals a2 plus d2 plus i2 plus c2, backslash display style r underscore mz equals a caret 2 plus d caret 2 plus I caret 2 plus c caret 2. RMZ equals a2 plus c2, backslash display style r underscore mz equals a caret 2 plus c caret 2. Dizygotic twin correlation, RDZ, backslash display style r underscore dz. RDZ equals 1 2 am2 plus 1 4 d2 plus k I2 plus c2, backslash display style r underscore dz equals backslash frac 1 2 a caret 2 plus backslash frac 1 4 d caret 2 plus k I caret 2 plus c caret 2. RDZ equals 1 2 am2 plus c2, backslash display style r underscore dz equals backslash frac 1 2 a caret 2 plus c caret 2, where k backslash display style k is an unknown, probably very small, quantity. The simplified Falconer formulation can then be used to derive estimates of a2 backslash display style a caret 2 c2 backslash display style c caret 2 and e2 backslash display style e caret 2. Rearranging and substituting the RMZ backslash display style r underscore mz and rdz backslash display style r underscore dz, equations. One can obtain an estimate of the additive genetic variance, or heritability, A2 equals 2, rmz rdz, backslash display style a caret, 2, equals 2, r underscore, mz, r underscore, dz. The non-shared environmental effect E2 equals 1 rmz, backslash display style e caret, 2, equals 1 r underscore, mz, and, finally, the shared environmental effect C2 equals 2 r dz rmz, backslash display style c caret, 2, equals 2 r underscore, dz, r underscore, mz. 26. The Falconer formulation is presented here to illustrate how the twin model works. Modern approaches use maximum likelihood to estimate the genetic and environmental variance components. 31. Measured genetic variance. Edit. The Human Genome Project has allowed scientists to directly genotype the sequence of human DNA nucleotides. 32. Once genotyped, genetic variance can be tested for association with a behavioral phenotype, such as mental disorder, cognitive ability, personality, and so on. 33. Candidate genes. One popular approach has been to test for association candidate genes with behavioral phenotypes, where the candidate gene is selected based on some a priori theory about biological mechanisms involved in the manifestation of a behavioral trait or phenotype. 34. In general, such studies have proven difficult to broadly replicate. 35, 36, 37, 38. And there has been concern raised that the false positive rate in this type of research is high. 34, 39. Genome-wide association studies. In genome-wide association studies, researchers test the relationship of millions of genetic polymorphisms with behavioral phenotypes across the genome. 33. This approach to genetic association studies is largely atheoretical, and typically not guided by a particular biological hypothesis regarding the phenotype. 33. Genetic association findings for behavioral traits and psychiatric disorders have been found to be highly polygenic, involving many small genetic effects. 40, 41, 42, 43, 44. Study results about which and to what degree various traits, IQ and language-related skills appear to be influenced by genetics. 45, 46. Genetic variants identified to be associated with some trait or disease through GWAS may be used to improve disease risk predictions. However, the genetic variants identified through GWAS of common genetic variants are most likely to have a modest effect on disease risk or development of a given trait. This is different from the strong genetic contribution seen in Mendelian conditions or for some rare variants that may have a larger effect on disease. SNP heritability and co-heritability. Recently, researchers have begun to use similarity between classically unrelated people at their measured single nucleotide polymorphisms, SNPs, to estimate genetic variation or covariation that is tagged by SNPs, using mixed effects models implemented in software such as genome-wide complex trait analysis, GCTA. 47, 48. To do this, researchers find the average genetic relatedness over all SNPs between all individuals in a, typically large, sample, and use Huysman-Elston regression or restricted maximum likelihood to estimate the genetic variation that is tagged by, or predicted by, the SNPs. The proportion of phenotypic variation that is accounted for by the genetic relatedness has been called SNP heritability. 49. Intuitively, SNP heritability increases to the degree that phenotypic similarity is predicted by genetic similarity at measured SNPs, and is expected to be lower than the true narrow-sense heritability to the degree that measured SNPs fail to tag, typically rare, causal variants. 50. The value of this method is that it is an independent way to estimate heritability that does not require the same assumptions as those in twin and family studies, and that it gives insight into the allelic frequency spectrum of the causal variants underlying trait variation. 51. Quasi-experimental designs. Edit. Some behavioral genetic designs are useful not to understand genetic influences on behavior, but to control for genetic influences to test environmentally mediated influences on behavior. 52. Such behavioral genetic designs may be considered a subset of natural experiments. 53. Quasi-experiments that attempt to take advantage of naturally occurring situations that mimic true experiments by providing some control over an independent variable. Natural experiments can be particularly useful when experiments are infeasible, due to practical or ethical limitations. 53. A general limitation of observational studies is that the relative influences of genes and environment are confounded. A simple demonstration of this fact is that measures of environmental influence are heritable. 54. Thus, observing a correlation between an environmental risk factor and a health outcome is not necessarily evidence for environmental influence on the health outcome. Similarly, in observational studies of parent-child behavioral transmission, for example, it is impossible to know if the transmission is due to genetic or environmental influences, due to the problem of passive gene-environment correlation. 53. The simple observation that the children of parents who use drugs are more likely to use drugs as adults does not indicate why the children are more likely to use drugs when they grow up. It could be because the children are modeling their parents' behavior. Equally plausible, it could be that the children inherited drug use predisposing genes from their parent, which put them at increased risk for drug use as adults regardless of their parents' behavior. Adoption studies, which parse the relative effects of rearing environment and genetic inheritance, find a small to negligible effect of rearing environment on smoking, alcohol, and marijuana use in adopted children. 55. Non-primary source needed, but a larger effect of rearing environment on harder drug use. 56. Non-primary source needed. Other behavioral genetic designs include discordant twin studies, 52, children of twins designs, 57, and Mendelian randomization. 58. General findings. Edit. There are many broad conclusions to be drawn from behavioral genetic research about the nature and origins of behavior. 59. Three major conclusions include SAD Smiley 3. All behavioral traits and disorders are influenced by genes. Environmental influences tend to make members of the same family more different, rather than more similar. The influence of genes tends to increase in relative importance as individuals age. Genetic influences on behavior are pervasive. Edit. It is clear from multiple lines of evidence that all researched behavioral traits and disorders are influenced by genes, that is, they are heritable. The single largest source of evidence comes from twin studies, where it is routinely observed that monozygotic, identical, twins are more similar to one another than are same-sex dizygotic, fraternal, twins. 12. The conclusion that genetic influences are pervasive has also been observed in research designs that do not depend on the assumptions of the twin method. Adoption studies show that adoptees are routinely more similar to their biological relatives than their adoptive relatives for a wide variety of traits and disorders. 3. In the Minnesota study of twins reared apart, monozygotic twins separated shortly after birth were reunited in adulthood. 60. These adopted, reared apart twins were as similar to one another as were twins reared together on a wide range of measures including general cognitive ability, personality, religious attitudes, and vocational interests, among others. 60. Approaches using genome-wide genotyping have allowed researchers to measure genetic relatedness between individuals and estimate heritability based on millions of genetic variants. Methods exist to test whether the extent of genetic similarity, aka relatedness, between nominally unrelated individuals who are not close or even distant relatives, is associated with phenotypic similarity. 48. Such methods do not rely on the same assumptions as twin or adoption studies, and routinely find evidence for heritability of behavioral traits and disorders. 42, 44, 61. Nature of environmental influence. Edit. Just as all researched human behavioral phenotypes are influenced by genes, i.e., are heritable, all such phenotypes are also influenced by the environment. 11, 59. The basic fact that monozygotic twins are genetically identical but are never perfectly concordant for psychiatric disorder or perfectly correlated for behavioral traits, indicates that the environment shapes human behavior. 59. The nature of this environmental influence, however, is such that it tends to make individuals in the same family more different from one another, not more similar to one another. 3. That is, estimates of shared environmental effects, C2, \C_2, in human studies are small, negligible, or zero for the vast majority of behavioral traits and psychiatric disorders, whereas estimates of non-shared environmental effects, E2, \E_2, are moderate to large. 11. From twin studies C2, \C_2, is typically estimated at 0 because the correlation, RMZ, \R_MZ, between monozygotic twins is at least twice the correlation, RDZ, \R_DZ, for dizygotic twins. When using the Falconer variance decomposition, 1.0 equals A2 plus C2 plus E2, \1.0 equals A2, plus C2, plus E2. This difference between monozygotic and dizygotic twin similarity results in an estimated C2 equals 0, \C2 equals 0. The Falconer decomposition is simplistic. 26. It removes the possible influence of dominance and epistatic effects which, if present, will tend to make monozygotic twins more similar than dizygotic twins and mask the influence of shared environmental effects. 26. This is a limitation of the twin design for estimating C2, \C2. However, the general conclusion that shared environmental effects are negligible does not rest on twin studies alone. Adoption research also fails to find large, C2, \C2, components, that is, adoptive parents and their adopted children tend to show much less resemblance to one another than the adopted child and his or her non-rearing biological parent. 3. In studies of adoptive families with at least one biological child and one adopted child, the sibling resemblance also tends to be nearly zero for most traits that have been studied. 11, 62. Similarity in twins and adoptees indicates a small role for shared environment in personality. The figure provides an example from personality research, where twin and adoption studies converge on the conclusion of zero to small influences of shared environment on broad personality traits measured by the multidimensional personality questionnaire including positive emotionality, negative emotionality, and constraint. 63. Given the conclusion that all researched behavioral traits and psychiatric disorders are heritable, biological siblings will always tend to be more similar to one another than will adopted siblings. However, For some traits, especially when measured during adolescence, adopted siblings do show some significant similarity, for example, correlations of 20 to one another. Traits that have been demonstrated to have significant shared environmental influences include internalizing and externalizing psychopathology, 64, substance use, 65, non-primary source needed, and dependence, 56, non-primary source needed, and intelligence, 65, non-primary source needed. Nature of genetic influence. Edit. Genetic effects on human behavioral outcomes can be described in multiple ways. 26. One way to describe the effect is in terms of how much variance in the behavior can be accounted for by alleles in the genetic variant, otherwise known as the coefficient of determination or R2, \r_2. An intuitive way to think about R2, \r_2, is that it describes the extent to which the genetic variant makes individuals, who harbor different alleles, different from one another on the behavioral outcome. A complementary way to describe effects of individual genetic variance is in how much change one expects on the behavioral outcome given a change in the number of risk alleles an individual harbors, often denoted by the Greek letter beta, \beta, denoting the slope in a regression equation, or, in the case of binary disease outcomes by the odds ratio OR, \or, of disease given allele status. Note the difference, R2, \r_2, describes the population level effect of alleles within a genetic variant, beta, \beta, or OR, \or, describe the effect of having a risk allele on the individual who harbors it, relative to an individual who does not harbor a risk allele. 66. When described on the R2, \r_2 metric, the effects of individual genetic variants on complex human behavioral traits and disorders are vanishingly small, with each variant accounting for R2 0.3% of variation in the phenotype. 3. This fact has been discovered primarily through genome-wide association studies of complex behavioral phenotypes, including results on substance use, 67, 68, personality, 69, fertility, 70, schizophrenia, 41, depression, 69, 71, and endophenotypes including brain structure, 72, and function. 73. There are a small handful of replicated and robustly studied exceptions to this rule, including the effect of APOE on Alzheimer's disease, 74, and Kerner-5 on smoking behavior, 67, and ALDH2, in individuals of East Asian ancestry, on alcohol use. 75. On the other hand, when assessing effects according to the beta, \beta metric, there are a large number of genetic variants that have very large effects on complex behavioral phenotypes. The risk alleles within such variants are exceedingly rare, such that their large behavioral effects impact only a small number of individuals. Thus, when assessed at a population level using the R2, \r_2 metric, they account for only a small amount of the differences in risk between individuals in the population. Examples include variants within AP that result in familial forms of severe early-onset Alzheimer's disease but affect only relatively few individuals. Compare this to risk alleles within APOE, which pose much smaller risk compared to AP, but are far more common and therefore affect a much greater proportion of the population. 76. Finally, there are classical behavioral disorders that are genetically simple in their etiology, such as Huntington's disease. Huntington's is caused by a single autosomal dominant variant in the HTT gene, which is the only variant that accounts for any differences among individuals in their risk for developing the disease, assuming they live long enough. 77. In the case of genetically simple and rare diseases such as Huntington's, the variant R2, /R2, and the OR, /or, are simultaneously large. 66. Additional general findings. Edit. In response to general concerns about the replicability of psychological research, behavioral geneticists Robert Plomin, John C. DeFries, Valerie Napick, and Jenny Niederheiser published a review of the 10 most well-replicated findings from behavioral genetics research. 59. The 10 findings were: all psychological traits show significant and substantial genetic influence. No behavioral traits are 100% heritable. Heritability is caused by many genes of small effect. Phenotypic correlations between psychological traits show significant and substantial genetic mediation. The heritability of intelligence increases throughout development. Age-to-age stability is mainly due to genetics. Most measures of the environment show significant genetic influence. Most associations between environmental measures and psychological traits are significantly mediated genetically. Most environmental effects are not shared by children growing up in the same family. Abnormal is normal. Criticisms and controversies. Edit. See also, missing heritability problem. Behavioral genetic research and findings have at times been controversial. Some of this controversy has arisen because behavioral genetic findings can challenge societal beliefs about the nature of human behavior and abilities. Major areas of controversy have included genetic research on topics such as racial differences, intelligence, violence, and human sexuality. 78. Other controversies have arisen due to misunderstandings of behavioral genetic research, whether by the lay public or the researchers themselves. 3. For example, the notion of heritability is easily misunderstood to imply causality, or that some behavior or condition is determined by one's genetic endowment. 79. When behavioral genetics researchers say that a behavior is X% heritable, that does not mean that genetics causes, determines, or fixes up to X% of the behavior. Instead, heritability is a statement about genetic differences correlated with trait differences on the population level. Citation needed. Historically, perhaps the most controversial subject has been on race and genetics. 78. Race is not a scientifically exact term, and its interpretation can depend on one's culture and country of origin. 80. Instead, geneticists use concepts such as ancestry, which is more rigorously defined. 81. For example, a so-called black race may include all individuals of relatively recent African descent, recent because all humans are descended from African ancestors. However, there is more genetic diversity in Africa than the rest of the world combined. 82. So speaking of a black race is without a precise genetic meaning. 81. Qualitative research has fostered arguments that behavioral genetics is an ungovernable field without scientific norms or consensus, which fosters controversy. The argument continues that this state of affairs has led to controversies including race, intelligence, instances where variation within a single gene was found to very strongly influence a controversial phenotype, for example, the gay gene controversy, and others. This argument further states that because of the persistence of controversy in behavior genetics and the failure of disputes to be resolved, behavior genetics does not conform to the standards of good science. 83. The scientific assumptions on which parts of behavioral genetic research are based have also been criticized as flawed. 79. Genome-wide association studies are often implemented with simplifying statistical assumptions, such as additivity, which may be statistically robust but unrealistic for some behaviors. Critics further contend that, in humans, behavior genetics represents a misguided form of genetic reductionism based on inaccurate interpretations of statistical analyses. 84. Studies comparing monozygotic, MZ, and dizygotic, DZ, twins assume that environmental influences will be the same in both types of twins, but this assumption may also be unrealistic. MZ twins may be treated more alike than DZ twins, 79, which itself may be an example of evocative gene-environment correlation, suggesting that one's genes influence their treatment by others. It is also not possible in twin studies to eliminate effects of the shared womb environment, although studies comparing twins who experience monochorionic and dichorionic environments in utero do exist, and indicate limited impact. 85. Studies of twins separated in early life include children who were separated not at birth but partway through childhood. 79. The effect of early rearing environment can therefore be evaluated to some extent in such a study, by comparing twin similarity for those twins separated early and those separated later. 60. See also. Edit. Behavior genetics. Behavior genetics association. Behavioral neurogenetics. Biocultural evolution. Evolutionary psychology. Genes, brain and behavior. Genome-wide association study. International Behavioral and Neural Genetics Society. International Society of Psychiatric Genetics. Journal of Neurogenetics. Nature versus Nurture. Personality Psychology. Psychiatric Genetics. Psychiatric Genetics. Quantitative Genetics. References. Edit. A.B. Lolin J.C., 2009. History of Behavior Genetics. In Kim Y., ed. Handbook of Behavior Genetics, 1 ed. New York, N.Y., Springer. pp. 311. Do 10.1007.978038777672777-1. ISBN 978-0-387-76726-0. A.B. Maxon S.C., 30 August 2006. A History of Behavior Genetics. In Jones B.C., Mohamed P., eds. Neurobehavioral Genetics, Methods and Applications, 2nd edition. CRC Press. ISBN 978-1-4200-03567. ABCDEFGHIJKLMNO McGUM, Gottesman 2, 2015. Behavior Genetics. The Encyclopedia of Clinical Psychology. pp. 111. Do 10.1002/97811862539 Beck 578. ISBN 97811862539. Vaughan v. Vaughan at, 1999. The Tempest. The Arden Shakespeare, 3rd ed. The Arden Shakespeare. p. 60. ISBN 978-1-903436-08-0. Hereditary Genius, An Inquiry into Its Laws and Consequences. London, Macmillan and Company 1869. Archived from the original on 7 December 2019. Retrieved 17 December 2009. Stigler S.M., July 2010. Darwin, Galton and the Statistical Enlightenment. Journal of the Royal Statistical Society, Series A 173,
3469482.10.1467-985x.2010.00643.x. S2 SID 533333238. Hall CS, 1951. The Genetics of Behavior. In Stevens S.S., ed. Handbook of Experimental Psychology. New York, John Wiley & Sons. pp. 304329. Grigorenko L., Ravikshkurbo I., 1997. Russian Psychogenetics. In Grigorenko L., ed. Psychology of Russia, Past, Present, Future. Comic, NY, Nova Science. pp. 83124. Broadhurst P.L., July 1969. Psychogenetics of Emotionality in the Rat. Annals of the New York Academy of Sciences. 159, 380624. Bibcode 1969 NYSA.159806B. Do 10.1000.1749-6632.1969.TB12980.x. PMID 5260300. S2 SID 423233956. Fuller J.L., Thomson W.R., 1960. Behavior Genetics. New York, John Wiley & Sons. ABCDE Podeman T.J., Benjamin B., de Leeuwca, Sullivan P.F., Van Botchaveno, Vischer P.M., et al. July 2015. Meta-analysis of the heritability of human traits based on 50 years of twin studies, PDF. Nature Genetics. 47, 77029 do 10.1038/ing.3285. PMID 259855137. S2 SID 205349969. Archived, PDF, from the original on 20 April 2022. Retrieved 30 January 2019. AB Turkheimer E. 2000. 3 Laws of Behavior Genetics and What They Mean, PDF. Current Directions in Psychological Science. 9. 516164 do 10.1467-8721.00084. S2 SID 2861437. Archived, PDF, from the original on 9 October 2016. Retrieved 12 April 2016. Eric Z, Selzam S, Smith Willie E, Napik V.S., Niederheiser J.M., De Vries J.C., et al. September 2016. Publication trends over 55 years of behavioral genetic research. Behavior Genetics. 46. 56037 do 10.1051/S1051-016-9786-2. PMC 5206393. PMID 269927331. AB Plomin R, De Vries J.C., Napik V.S., Niederheiser M., 24 September 2012. Behavioral Genetics. Worth Publishers.
ISBN 978-1-4292-4215-8. Archived from the original on 31 December 2013. Retrieved 27 January 2016. Plomin A. Behavior Genetics. Encyclopaedia Britannica. Archived from the original on 17 April 2021. Retrieved 15 June 2018. De Vries J.C., Hegman J.P., Halcombe Ra., August 1974. Response to 20 generations of selection for open field activity in mice. Behavioral Biology. 11. 448195-10.1016/S0091-6773, 74, 90800-1. PMID 44155597. Lynch C.B., November 1980. Response to divergent selection for nesting behavior in Mus musculus. Genetics. 96, 375765-10.1093/genetics/96.3.757. PMC 1214374. PMID 7196362. Swallow J.G., Carter Pa, Garland T., May 1998. Artificial selection for increased wheel-running behavior in house mice. Behavior Genetics. 28, 322737-10.1023/10214793317779. PMID 9670598. S2 SID 183362443. Heidenreich M., Zhang F., January 2016. Applications of CRISPR-Cas systems in neuroscience. Nature Reviews. Neuroscience. 17, 136-44-10.1038/NRN.2015.2. PMC 4899966. PMID 266565253. Singh P., Skymenti J.C., Bolkunfilis E., January 2015. A Mouse Geneticist's Practical Guide to CRISPR Applications. Genetics. 199, 1110.1034/genetics.114.1697771. PMC 4286675. PMID 252713004. Cryan J.F., Holmes A., September 2005. The Ascent of Mouse, Advances in Modeling Human Depression and Anxiety. Nature Reviews. Drug Discovery. 4, 9775790.10.1038/NRD1825. PMID 161381008. S2 SID 1820734. Woman M., Granato M., March 2012. Behavioral Genetics in Larval Zebrafish, Learning from the Young. Developmental Neurobiology. 72, 3366372.10.1002/GNU.20872. PMC 6430578. PMID 223282773. and Holt R., Mackay T.F., 1 April 2015. Dissecting the Genetic Architecture of Behavior in Drosophila melanogaster. Current Opinion in Behavioral Sciences. 2, 17. do 10.1016/j.kbh.2014.06.001 PMC 4507818 PMID 262034060 Walensky E. Wei J. March 1990. The Behavioral Genetics of Caenorhabditis elegans. Behavior Genetics. 20.216918 do 10.1007/BF01067789 PMID 2191646 S2 SID 2371967 Starshare Lee C.N., Wang E., Ferris E., Bennett K., Steinwand S., Rhodson, et al. August 2019. Complex economic behavior patterns are constructed from finite, genetically controlled modules of behavior. Cell reports. 28. 7.18.14.18.29.E6 to 10.1016/j.cellrip.2019.07.038 PMC 7476553 PMID 314122249 S2 SID 199662477 ABCDEFGH Falconer DS, 1989. Introduction to Quantitative Genetics. Longman, Scientific and Technical. ISBN 978-0-470-21162-5. Archived from the original on the 22nd of April, 2021. Retrieved the 2nd of December, 2016. Eves L., Foley D., Silberg J., 2003. Has the equal environments assumption been tested in twin studies? Twin Research. 6.64869 do 10.1369052032033226864743 PMID 149654558 Kendall K.S., Neil M.C., Kessler R.C., Heath A.C., Eves L.J., January 1993. A test of the equal environment assumption in twin studies of psychiatric illness. Behavior Genetics. 23, 121-7. Cite CX 10.1.1.595.7413. Do 10.1007/BF01067551. PMID 8476388. S2 SID 9034050. Harden KP, the 21st of September 2021. The Genetic Lottery: Why DNA Matters for Social Equality. Princeton University Press. p. 276. ISBN 978-0-691-22670-5. Jinks J.L., Fuka D.W., 1970. Comparison of the biometrical genetical, MAVAT, and classical approaches to the analysis of the human behavior. Psychological Bulletin. 73. 5311349 to 10.1037/H002935. PMID 5528333. Martin Ing, Eves L.J., February 1977. The Genetical Analysis of Covariance Structure. Heredity. 38. 179.95 to 10.1038/HDY.1977.9. PMID 268313. Lander S., February 2011. Initial impact of the sequencing of the human genome. Nature. 470, 7333 18797. Bibcode 2011 Nature.47187L. Do 10.1038 / Nature 09792. HDL 1721.1 / 69154. PMID 2130791. S2 SID 4344403. ABC McCarthy Me, Abecassis GR, Carden LR, Goldstein DB, Little J, Ionides JP, IT AL. May 2008. Genome-wide association studies for complex traits, consensus, uncertainty and challenges. Nature Reviews Genetics. 9. 53569 do 10.1038 / NRG 2344. PMID 1839818. S2 SID 15032294. AB Duncan Lowe, Keller MC, October 2011. A critical review of the first 10 years of candidate gene by environment interaction research in psychiatry. The American Journal of Psychiatry. 168, 10 1041 9 do 10.1176 / appy.archb.2011.11020191. PMC 3222234. PMID 2189071. Farrell MS, Werge T, Sklar P, Owen MJ, Ofofra, O'Donovan MC, IT AL. May 2015. Evaluating historical candidate genes for schizophrenia. Molecular Psychiatry. 20. 5555562 do 10.1038 / MP.2015.16. PMC 4414705. PMID 257548081. Hewitt J.K., January 2012. Editorial Policy on Candidate Gene Association and Candidate Gene by Environment Interaction Studies of Complex Traits. Behavior Genetics. 42, 112 do 10.1007 / S10519-011-9504Z. PMID 219280046. S2 SID 11492871. Johnson EC, Border R, Melroy Grefwe, de LEUWCA, A. Ringamar, Keller MC, November 2017. No evidence that schizophrenia candidate genes are more associated with schizophrenia than non-candidate genes. Biological Psychiatry. Risk genes and the emergence of schizophrenia. 82, 10702708 do 10.1016 / j.biopsych.2017.06.033. PMC 5643230. PMID 2882370. Border R, Johnson EC, Evans LM, Smolano, Bully N, Sullivan PF, IT AL. May 2019. No support for historical candidate gene or candidate gene by interaction hypotheses for major depression across multiple large samples. The American Journal of Psychiatry. 176, 5376387 do 10.1176 / appy.archb.2018.180708881. PMC 6548317. PMID 3084580. Colhoun HM, McKeague PM, Davy Smith G, March 2003. Problems of reporting genetic associations with complex outcomes. Lancet. 361, 9368 65872 to 10.1016/S0140-6736, 03, 12715-8. PMID 126420066. S2 SID 156795661. Vischer PM, Brown Ma, McCarthy Me, Yang J., January 2012. 5 Years of Bus Discovery. American Journal of Human Genetics. 90, 1724 to 10.1016/J.H.2011.11.029. PMC 3257326. PMID 222439664. AB Ripkes, Neil B. M., Corvino, Walters J. T., Farr K. H., Holmans Pa, et al. Schizophrenia Working Group of the Psychiatric Genomics Consortium, July 2014. Biological Insights from 108 Schizophrenia-Associated Genetic Loci. Nature. 511, 7510 4217. Bibcode 2014 Nature.511421s. Do 10.1038/Nature13595. PMC 4112379. PMID 250566061. AB Leisha, De Candia T.R., Ripkes, Yang J., Sullivan P.F., Goddardme, et al. February 2012. Estimating the proportion of variation in susceptibility to schizophrenia captured by common SNPs. Nature Genetics. 44, 324750 to 10.1038/ing.1108. PMC 3327879. PMID 223442220. Sullivan P.F., Daly M.J., O'Donovan M., July 2012. Genetic Architectures of Psychiatric Disorders, The Emerging Picture and Its Implications. Nature Reviews. Genetics. 13, 853751 to 10.1038/NRG 3240. PMC 4110909. PMID 2277727. AB de Moor M.H., Van den Berg S.M., Verwijch K.J., Kruger R.F., Luciano M., Arias Vasquez, et al. July 2015. Meta-analysis of genome-wide association studies for neuroticism, and the polygenic association with major depressive disorder. JAMA Psychiatry. 72, 764-250 to 10.1011/Jamapsychiatry.2015.0554. PMC 4667957. PMID 259936007. Massive Genome Study informs the biology of reading and language. Max Planck Society via medicalexpress.com. Retrieved 18 September 2022. Ising Mirza Schreiber N. De ZL, Wang CA, Trung DT, Allegrini AG, et al. August 2022. Genome-wide analyses of individual differences in quantitatively assessed reading and language-related skills in up to 34,000 people. Proceedings of the National Academy of Sciences of the United States of America. 119, 35 E22027641119. Bibcode 2022 NAS 119027664E. Do 10.1073/NAS.22027641119. PMC 9436320. PMID 359982220. Carrot Yong J, Benjamin B, McAvoy BP, Gordon S, Henders AK, Nihold Dr, et al. July 2010. Common SNPs explain a large proportion of the heritability for human height. Nature Genetics. 42, 75659 to 10.1038/ing.608. PMC 3232052. PMID 20562875. Carrot AB Yong J, Leisha, Goddardme, Visha PM, January 2011. GCTA, a tool for genome-wide complex trait analysis. American Journal of Human Genetics. 88, 17682 to 10.1016/J.H.2010.11.011. PMC 3014363. PMID 2116748. Carrot Leisha, Yong J, Chen GB, Ripkes, Still EA, Hultman cm, et al. 2013. Estimation of SNP heritability from dense genotype data. American Journal of Human Genetics. 93, 61151 5 to 10.1016/J.H.2013.10.015. PMC 3852919. PMID 2431450. Carrot Visha PM, Yong J, Goddardme, 2010. A commentary on common SNPs explain a large proportion of the heritability for human height by Yong et al. 2010. Twin Research and Human Genetics. 13, 651724 to 10.1375/twin.13.6.517. PMID 211422928. S2 SID 157309555. Carrot Ray NR, Leisha, Meta D, Vinkhuizen A, Dudbridge F, Middeldorp cm, 2014. Research Review, Polygenic Methods and Their Application to Psychiatric Traits, PDF. Journal of Child Psychology and Psychiatry, and Allied Disciplines. 55, 10.106887 to 10.1111/JCPP.12295. PMID 2513240. Archived, PDF, from the original on 16 January 2017. Retrieved 1 July 2019. Carrot AB McGew M, Osler M, Christensen K, September 2010. Causal Inference and Observational Research, The Utility of Twins. Perspectives on Psychological Science. 5. 554656 to 10.11177/17456916103835511. PMC 3094752. PMID 215938989. Carrot ABC Rutter M, December 2007. Proceeding from observed correlation to causal inference, the use of natural experiments. Perspectives on Psychological Science. 2, 437795. Site CX 10.1.1.649.2804. do 10.1745-6916.2007.00050.x. PMID 26151974. S2 SID 2059081149. Kendall K.S., Baker J.H., May 2007. Genetic Influences on Measures of the Environment, A Systematic Review. Psychological Medicine. 37. 561526 to 10.1017/S00332917060095524, inactive 1 November 2024. PMID 17176502. S2 SID 435981444. Site Journal, CS1 maint. DOI inactive as of November 2024. Link. Keys M, Legrand LN, Iacono WG, McGew M, October 2008. Parental Smoking and Adolescent Problem Behavior, an Adoption Study of General and Specific Effects. The American Journal of Psychiatry. 165. 10.1338 do 10.1176/appy.archb.2008.080100125. PMC 2597022. PMID 186765889. AB Kendall K.S., Sundquist K., Olson H., Palmer K., Mars H., Winkleby Ma, et al. July 2012. Genetic and Familial Environmental Influences on the Risk for Drug Abuse, a National Swedish Adoption Study. Archives of General Psychiatry. 69. 7697 to 10.1001/archgentpsychiatry.2011.2112. PMC 3556483. PMID 223932006. D'Onofrio BM, Turkheimer EN, Eves LJ, Corey LA, Berg K, Solas MH, et al. November 2003. The Role of the Children of Twins Design in Elucidating Causal Relations Between Parent Characteristics and Child Outcomes. Journal of Child Psychology and Psychiatry, and Allied Disciplines. 44, 81130 44 do 10.1169/7610.00196. PMID 146264555. Smith G.D., Ibrahim S., February 2004. Mendelian Randomization, Prospects, Potentials, and Limitations. International Journal of Epidemiology. 33, 13042 do 10.1093/ig/DYH132. PMID 15075143. ABCD Plomin R. De Vries J.C., Napik V.S., Niederheiser J.M., January 2016. Top 10 Replicated Findings from Behavioral Genetics. Perspectives on Psychological Science. 11. 1. Published 27 January 2016 323 do 10.11456916156156174339. PMC 4739500. PMID 268177721. ABC Bouchard T.J., Likon D.T., McGew M., Siegel N.L., Telegeno, October 1990. Sources of human psychological differences, the Minnesota study of twins reared apart. Science. 250, 4978-2238. Bibcode 1990 SCI, 25223B. Site CX10.1.1.225.1769. Do 10.1026/science.2218526. PMID 2218526. S2 SID 1179469. Carrot Plomin R, Hayworth cm, Meeburn L, Price T.S., Davis O.S., April 2013. Common DNA markers can account for more than half of the genetic influence on cognitive abilities. Psychological Science. 24, 45628 2:4.10.101177/0956797612457952. PMC 3652710. PMID 2350197. Carrot Plomin R, Daniels D., June 2011. Why are children in the same family so different from one another? International Journal of Epidemiology. 40, 356382 do 10.1093/ige/dyq148. PMC 3147063. PMID 218076442. Carrot Matson L.K., McGew M., Iacono W.G., November 2013. Shared Environmental Influences on Personality, a Combined Twin and Adoption Approach. Behavior Genetics. 43, 6491504 do 10.100507-4-19509-013-9616-8. PMC 3868213. PMID 240655564. Carrot Burt Sa., July 2009. Rethinking Environmental Contributions to Child and Adolescent Psychopathology, a Meta-Analysis of Shared Environmental Influences. Psychological Bulletin. 135, 460837 do 10.1037/0015702. PMID 195866164. Carrot A.B. Buchanan J.P., McGew M., Keyes M., Iacono W.G., September 2009. Are there shared environmental influences on adolescent behavior? Evidence from a study of adoptive siblings. Behavior Genetics. 39, 55340 do 10.1057/10519-009-9283 PMC 2858574. PMID 196264434. Carrot A.B. Bland J.M., Altman D.G., May 2000. Statistics Notes. The odds ratio. BMJ. 320, 7,247 1,468 to 10.1036/BMJ.320.700047.1468. PMC 11276551. PMID 108270661. Carrot A.B. Thorgerson Te, Gudbjornsson D.F., Surakar I, Vink J.M., Armin N., Geller F., et al. May 2010. Sequence variants at CHRNB3 Kerner 6 and CYP2A6 affect smoking behavior. Nature Genetics. 42, 544853 do 10.1038/ing.573. PMC 30806000. PMID 20418888. Skuman G, Coin LJ, Ilyaduzamia, Shiro NP, Berger KH, Stacy D, It al. April 2011. Genome-wide association and genetic functional studies identify autism susceptibility candidate 2 gene, AUTS2, in the regulation of alcohol consumption. Proceedings of the National Academy of Sciences of the United States of America. 108, 17 7119 24. Bib code 2011 as 108.7119s. Do 10.1073/NAS.1017288108. PMC 30840048. PMID 21471458. AB Oak Bayer, Barcelmans BM, Deneve JE, Turley P, Nevar MG, Fontana Ma, It al. June 2016. Genetic variants associated with subjective well-being, depressive symptoms, and neuroticism identified through genome-wide analyses. Nature Genetics. 48, 662433 do 10.1038/ing.3552. PMC 48845152. PMID 27089181. Day FR, Helgeson H, Shazman DI, Rose LM, Lopi R, Scott Ra, It al. June 2016. Physical and neurobehavioral determinants of reproductive onset and success. Nature Genetics. 48, 661723 do 10.1038/ing.3551. PMC 52388953. PMID 27089180. Converge Consortium, July 2015. Sparse whole genome sequencing identifies 2 loci for major depressive disorder. Nature. 523, 7562 58891. Bib code 2015 Nature.523588C. Do 10.1038/Nature14659. PMC 45222619. PMID 26176920. High bar DP, Stein JL, Rentierium Me, Arias Vasquez, Des Rivières S, Chahan Shad N, It al. April 2015. Common genetic variants influence human subcortical brain structures. Nature. 520, 7546 2249. Bib code 2015 Nature.52224. Do 10.1038/Nature14101. PMC 43933666. PMID 2560738. Iacono WG, Vaidyanathon U, Vries Sci, Malone SM, December 2014. Knowns and unknowns for psychophysiological endophenotypes, integration and response to commentaries. Psychophysiology. 51, 12 1339 47 do 10.1111/PSYP.12358. PMC 42314888. PMID 25387720. Corda A, Saundersm, Risk NJ, Strit Matter WJ, Schmickeldo, Gaskell PC, It al. June 1994. Protective effect of apolipoprotein E type 2 allele for late onset Alzheimer's disease. Nature Genetics. 7. 2184 do 10.1038/0694-180. PMID 79200638. S2 SID 11137478. Luchak SE, Glatt SJ, Wall TL, July 2006. Meta-analyses of ALDH2 and ADH1B with alcohol dependence in Asians. Psychological Bulletin. 132, 460721 do 10.1037/0033-2909.132.4.607. PMID 16822169. Guerrero RJ, Gustafsson DR, Hardy J, March 2012. The Genetic Architecture of Alzheimer's Disease, Beyond AP, PSENs and APOE. Neurobiology of Aging. 33, 343756 do 10.1016/j.neurobiolaging.2010.03.025. PMC 2980860. PMID 2059461. Gasella JF, Wexler NS, Keneally PM, Naylor SL, Anderson Ma, Tanziri, it al. 1983. A polymorphic DNA marker genetically linked to Huntington's disease. Nature. 306, 5942 348. Bibcode 1983 Nature.306234G. Do 10.1038/30623440. PMID 6316146. S2 SID 43200711. AB Hayden EC, October 2013. Ethics, Taboo Genetics. Nature. 502, 7469 26-8. Bibcode 2013 Nature.502, 26C. Do 10.1038/502026A. PMID 240919664. ABCD Charney E, January 2017. Genes, Behavior, and Behavior Genetics. Highly Interdisciplinary Reviews, Cognitive Science. 8, 12E 1405 do 10.1002/WCS.1405 HDL 10161/13337. PMID 2790659. Udall M, Roberts D, DeSalle R, Tishkoff S, February 2016. Science and Society, Taking Race Out of Human Genetics. Science. 351, 6273 5645. Bibcode 2016 SCI, 351564Y. Do 10.1126/science.aac 4951. PMID 2691260. S2 SID 2066393006. AB BRYCK, Duran DY, McPherson JM, Reek D, Mountain JL, January 2015. The Genetic Ancestry of African Americans, Latinos, and European Americans across the United States. American Journal of Human Genetics. 96, 13753 do 10.1016/j.j.2014.11.010 PMC 4289685 PMID 255296636 Caritabecassis GR, Ortano, Brooks LD, De Pristo Ma, Durbin RM, Hand Sakari, IT al. November 2012. An integrated map of genetic variation from 1092 human genomes. Nature. 491, 7422 5665. Bibcode 2012 Nature.491, 56T. Do 10.1038/Nature11632. PMC 3498066. PMID 231282226. Karit Panofskia, the 7th of July 2014. Misbehaving Science, Controversy and the Development of Behavior Genetics. University of Chicago Press. ISBN 978-0-226-05859-7. Archived from the original on the 22nd of April 2021. Retrieved the 23rd of May 2018. Karit Lerner RM, the 27th of August 2015. Eliminating Genetic Reductionism from Developmental Science. Research in Human Development. 12. 3417818 to 10.1080.2015.1068058. ISSN 1542-7609. S2 SID 143195504. Karit van Beijsterveld CE, Overbeek Lee, Rosendahl L, McMaster Mount, Glasner TJ, Bartels M, IT al. May 2016. Chorionicity and heritability estimates from twin studies, the prenatal environment of twins and their resemblance across a large number of traits. Behavior Genetics. 46. 330-414 to 10.1007/105-9745-3. PMC 4858554. PMID 2641067. Further reading. Edit. Krujowy, 2015. Key Issues in Contemporary Behavioral Genetics. Current Opinion in Behavioral Sciences. 2, 8995. Do 10.1016/j.kbh.2014.10.002. S2 SID 532000512. Krujowy, JLIRT, eds. 1999. Handbook of Molecular Genetic Techniques for Brain and Behavior Research. Techniques in the Behavioral and Neural Sciences. Volume 13. Elsevier. ISBN 978-0-444-50239-1. Krujowy, Sluiter F., JLIRT, Pietropaulo S., eds. 2013. Behavioral Genetics of the Mouse, Genetics of Behavioral Phenotypes. Cambridge Handbooks in Behavioral Genetics. Volume 1. Cambridge University Press. ISBN 978-1-107-03481-5. Flint J., Greenspan R.J., Kendall K.S., the 28th of January 2010. How Genes Influence Behavior. Oxford University Press. ISBN 978-0-19-955990-9. Johnson W., Turkheimer E., Gottesman II, Bouchard T.J., August 2010. Beyond Heritability, Twin Studies in Behavioral Research. Current Directions in Psychological Science. 18. 42171.10.1467-8767-8067-2009.01639.x. PMC 2899491. PMID 206-25474. Johnson W., Penkel, Spinath FM, 2011. Understanding Heritability, What It Is and What It Is Not, PDF. European Journal of Personality. 25. 4287294-10.1002/per.835. ISSN 0890-2070. S2 SID 4184245. Archived, PDF, from the original on 16 December 2013. Retrieved 15 December 2013. Maxon SC, 10 October 2012. Chapter 1, Behavioral Genetics. In Wiener IB, Nelson R.J., Mitsumari S., eds. Handbook of Psychology, Volume 3, Behavioral Neuroscience. John Wiley and Sons. ISBN 978-0-470-89059-2. Archived from the original on 16 December 2013. Retrieved 15 December 2013. Spinath FM, Johnson W., 2011. Chapter 10, Behavior Genetics. In Chamorro Pre-Music T. von Stum S., Fernhammer, eds. The Huyler Blackwell Handbook of Individual Differences. United Kingdom, Blackwell Publishing Limited to 10.1002/9781444343120. ISBN 978-1-4443-3438-8. External links. Edit. Wikimedia Commons has media related to behavioral genetics. McGew M., 5 May 2014. Introduction to Human Behavioral Genetics. Coursera. Retrieved 10 June 2014. McGew M., December 2020. Introduction to Behavioral Genetics. University of Minnesota. Retrieved 28 June 2021. Behavior Genetics Association. Behavior Genetics Association. Retrieved 8 May 2022. Institute for Behavioral Genetics at the University of Colorado Boulder, University of Colorado Boulder. Virginia Institute for Psychiatric and Behavioral Genetics. Virginia Commonwealth University. V. T. E. Genetics. Introduction. Outline. History. Timeline. Index. Glossary. Key components. Chromosome. DNA. RNA. Genome. Heredity. Nucleotide. Mutation. Genetic variation. Allele. Amino acid. Fields. Classical. Conservation. Cytogenetics. Ecological. Immunogenetics. Microbial. Molecular. Population. Quantitative. Archaeogenetics of. Africa, the Americas, the British Isles, Europe, Italy, the Middle East, South Asia. Related topics: behavioral genetics, epigenetics, geneticist, genome editing, genomics, genetic code, genetic engineering, genetic diversity, genetic monitoring, genetic genealogy, heredity, hygiene affair, medical genetics, missing heritability problem, molecular evolution, plant genetics, population genomics, reverse genetics, lists, list of genetic codes, list of genetics research organizations, category, V, T, E, neuroscience, outline, history, basic, science, behavioral epigenetics, behavioral genetics, brain mapping, brain reading, cellular neuroscience, computational neuroscience, connectomics, imaging genetics, integrative neuroscience, molecular neuroscience, neural decoding, neural engineering, neuroanatomy, neurobiology, neurochemistry, neuroendocrinology, neurogenetics, neuroinformatics, neurometrics, neuromorphology, neurophysics, neurophysiology, systems neuroscience, clinical neuroscience, behavioral neurology, clinical neurophysiology, epileptology, neurocardiology, neuroepidemiology, neurogastroenterology, neuroimmunology, neurointensive care, neurology, neurooncology, neuroophthalmology, neuropathology, neuropharmacology, neuroprosthetics, neuropsychiatry, neuroradiology, neurorehabilitation, neurosurgery, neurotology, neurovirology, nutritional neuroscience, psychiatry, cognitive neuroscience, affective neuroscience, behavioral neuroscience, chronobiology, molecular cellular cognition, motor control, neurolinguistics, neuropsychology, sensory neuroscience, social cognitive neuroscience, interdisciplinary fields, consumer neuroscience, cultural neuroscience, educational neuroscience, evolutionary neuroscience, global neurosurgery, neuroanthropology, neural engineering, neurobiotics, neurocriminology, neuroeconomics, neuroepistemology, neuroaesthetics, neuroethics, neuroethology, neurohistory, neurolaw, neuromarketing, neuromorphic engineering, neurophenomenology, neurophilosophy, neuropolitics, neurorobotics, neurotheology, paleoneurobiology, social neuroscience, concepts, brain-computer interface, development of the nervous system, neural network, artificial, neural network, biological, detection theory, intraoperative neurophysiological monitoring, neurochip, neurodegenerative disease, neurodevelopmental disorder, neurodiversity, neurogenesis, neuroimaging, neuroimmune system, neuromanagement, neuromodulation, neuroplasticity, neurotechnology, neurotoxin, self-awareness, category, commons, v, t, e, psychology, history, philosophy, portal, psychologist, basic, psychology, abnormal, affective neuroscience, affective science, behavioral genetics, behavioral neuroscience, behaviorism, cognitive/cognitivism, cognitive neuroscience, social, comparative, cross-cultural, cultural, developmental, differential, ecological, evolutionary, experimental, gestalt, intelligence, mathematical, moral, neuropsychology, perception, personality, psycholinguistics, psychophysiology, quantitative, social, theoretical, applied, psychology, anomalistic, applied behavior analysis, assessment, clinical, coaching, community, consumer, counseling, critical, educational, ergonomics, feminist, forensic, health, humanistic, industrial and organizational, legal, media, medical, military, music, occupational health, pastoral, political, positive, psychometrics, psychotherapy, religion, school, sport and exercise, suicidology, systems, traffic, methodologies, animal testing, archival research, behavior epigenetics, case study, content analysis, experiments, human subject research, interviews, neuroimaging, observation, psychophysics, qualitative research, quantitative research, self-report inventory, statistical surveys, concepts, behavior, behavioral engineering, behavioral genetics, behavioral neuroscience, cognition, competence, consciousness, consumer behavior, emotions, feelings, human factors and ergonomics, intelligence, mind, psychology of religion, psychometrics, terror management theory, psychologists, Wilhelm Wundt, William James, Ivan Pavlov, Sigmund Freud, Edward Thorndike, Carl Jung, John B. Watson, Clark L. Hull, Kurt Lewin, Jean Piaget, Gordon Allport, J. P. Guilford, Carl Rogers, Eric Erikson, B. F. Skinner, Donald O. Hebb, Ernest Hilgard, Harry Harlow, Raymond Cattell, Abraham Maslow, Neil E. Miller, Jerome Bruner, Donald T. Campbell, Hans Eysenck, Herbert A. Simon, David McClelland, Leon Festinger, Georgia Miller, Richard Lazarus, Stanley Scudder, Robert Zajanc, Albert Bandura, Roger Brown, Endel Tulving, Lawrence Kohlberg, Noam Chomsky, Ulrich Nieser, Jerome Keegan, Walter Mischel, Elliot Aronson, Daniel Kahneman, Paul Ekman, Michael Posner, Amos Tversky, Bruce McEwen, Larry Squire, Richard E. Nisbet, Martin Seligman, Ed D. Enner, Shelley E. Taylor, John Anderson, Ronald C. Kessler, Joseph E. Ladous, Richard Davidson, Susan Fiske, Roy Baumeister. Lists. Counseling topics. Disciplines. Organizations. Outline. Psychologists. Psychotherapies. Research methods. Schools of thought. Timeline. Topics. Wiktionary definition. Wiktionary category. Wikisource. Wikimedia Commons. Wikiquote. Wikinews. Wikibooks. V. T. E. Ethology. Branches. Animal cognition. Animal communication. Animal consciousness. Animal culture. Animal sexual behavior. Animal welfare science. Anthrozoology. B-learning and communication. Behavioral ecology. Behavioral endocrinology. Behavioral genetics. Breed. Cognitive ethology. Comfort behavior. Grooming. Comparative psychology. Emotion in animals. Ethogram. Evolutionary neuroscience. Feeding. Hover. Human ethology. Instinct. Learning. Neuroethology. Pain in animals. Philosophical ethology. Sociobiology. Stereotypy. Structures. Hive. Honeycomb. Nest. Instinct. Swarm. Tool used by non-humans. Zoosmiotics. Zoomusicology. Ethologists. Patrick Bateson. Mark Beckhoff. Donald Broom. John B. Calhoun. Charles Darwin. Marion Dawkins. Richard Dawkins. Irene Orzibler Bisfeld. Diane Fossey. Carl von Frisk. Jane Goodall. Heine Heidegger. Julian Huxley. Konrad Lorenz. Desmond Morris. Thomas Sabirk. William Homenthorpe. Nicolas Tinbergen. Jacob von Uexküll. Wolfgang Wickler. E. O. Wilson. Solly Zuckerman. Societies. Association for the Study of Animal Behavior. International Society for Applied Ethology. Journals. Animal Behavior. Animal Cognition. Animal Welfare. Behavioral Ecology. Behavior. Category. V. T. E. Evolutionary Psychology. History. Evolutionary Thought. Theoretical Foundations. Adaptationism. Cognitive Revolution. Cognitivism. Gene Selection Theory. Modern Synthesis. Criticism. Evolutionary. Processes. Adaptations. Altruism. Cheating. Hamiltonian spite. Reciprocal. Baldwin effect. Byproducts. Evolutionarily stable strategy. Exaptation. Fitness. Inclusive. Kin selection. Mismatch. Natural selection. Parental investment. Parent-offspring conflict. Sexual selection. Costly signaling. Male/female intrasexual competition. mate choice, sexual dimorphism, social selection, areas, cognition / emotion, affect, display rules, facial expression, behavioral modernity, cognitive module / modularity of mind, automatic and controlled processes, computational theory of mind, domain generality, domain specificity, dual process theory, cognitive trade-off hypothesis, evolution of the brain, evolution of nervous systems, fight or flight response, arachnophobia, bassophobia, ophidiophobia, folk biology / taxonomy, folk psychology / theory of mind, intelligence, Flynn effect, Wason selection task, motor control / skill, multitasking, sleep, visual perception, color vision, eye, naive physics, culture, aesthetics, literary criticism, musicology, anthropology, biological, crime, language, origin, psychology, speech, morality, moral foundations, religion, origin, universals, development, attachment, bonding, affectional / maternal / paternal bond, caregiver deprivation, childhood attachment, Cinderella effect, cognitive development, education, language acquisition, personality development, socialization, human factors / mental health, cognitive ergonomics, computer-mediated communication, engineering psychology, human-computer interaction, media naturalness theory, neuroergonomics, depression, digital media use and mental health, hyperphobia, imprinted brain hypothesis, mind blindness, psychological effects of internet use, rank theory of depression, schizophrenia, screen time, smartphones and pedestrian safety, social aspects of television, societal impacts of cars, distracted driving, lead crime hypothesis, mobile phones and driving safety, texting while driving, sex, activity, adult attachment, age disparity, arousal, concealed ovulation, Coolidge effect, desire, fantasy, hormonal motivation, jealousy, mate guarding, mating preferences, mating strategies, orientation, ovulatory shift hypothesis, pair bond, physical / sexual attraction, sexuality / male / female, sexy sun hypothesis, Westermarck effect, sex differences, aggression, autism, cognition, crime, division of labor, emotional intelligence, empathizing systemicing theory, gender role, intelligence, memory, mental health, narcissism, neuroscience, schizophrenia, substance abuse, suicide, variability hypothesis, related subjects, academic disciplines, behavioral / evolutionary economics, behavioral epigenetics / genetics, affective/behavioral/cognitive/evolutionary neuroscience, biocultural anthropology, biological psychiatry, cognitive psychology, cognitive science, cross-cultural psychology, ethology, evolutionary biology, evolutionary medicine, functional psychology, neuropsychology, philosophy of mind, population genetics, primatology, sociobiology, research topics, cultural evolution, evolutionary epistemology, great ape language, human-animal communication, missing heritability problem, primate cognition, unit of selection, coevolution, cultural group selection, dual inheritance theory, Fisher's principle, group selection, hologenome theory, Lamarckism, population, punctuated equilibrium, recent human evolution, species, species complex, transgenerational epigenetic inheritance, Trivers-Willard hypothesis, theoretical positions, cultural selection theory, determinism/indeterminism, biological determinism, connectionism, cultural determinism, environmental determinism, nature versus nurture, psychological nativism, social constructionism, social determinism, standard social science model, functionalism, memetics, multilineal evolution, neo-Darwinism, neo-evolutionism, socio-cultural evolution, unilineal evolution, evolutionary psychologists, evolutionary psychology, psychology portal, evolutionary biology portal, authority control databases, national, Germany, United States, Czech Republic, Israel. Retrieved from https://en.wikipedia.org/w/index.php title =behavioral_genetics=12593000081 Category: Behavioral genetics. Hidden categories. CS1 maint. DOI inactive as of November 2024. Articles with short description. Short description is different from Wikidata. Articles with excerpts. Good articles. Use British English from August 2015. Articles to be expanded from February 2020. All articles to be expanded. All pages needing factual verification. Wikipedia articles needing factual verification from February 2024. All articles with unsourced statements. Articles with unsourced statements from June 2020. Commons category link from Wikidata. This page was last edited on the 24th of November 2024 at 11:54 UTC. Text is available under the Creative Commons Attribution Share Alike 4.0 license. Additional terms may apply. By using this site, you agree to the terms of use and privacy policy. Wikipedia registered trademark symbol is a registered trademark of the Wikimedia Foundation, Inc., a non-profit organization. Privacy policy. About Wikipedia. Disclaimers. Contact Wikipedia. Code of conduct. Developers. Statistics. Cookie statement. Mobile view.